Hello students, this is Gagan. Welcome to GIPS Biology. Today we are going to talk about a topic called gluconeogenesis, which is a very important topic for life science students as well. So I am able to cover this topic within 15 minutes so that we can able to understand this topic very deeply in a depth manner itself. So first of all, we have to see what is the definition and introduction of gluconeogenesis. The name itself indicates that gluco means glucose, neo means new, and genesis means formation or production. So here we are preparing the glucose from a non-carbohydrate precursor, that is new precursor other than the carbohydrate molecule. So the precursors may be lactate, pyruvate, propionate, and many other amino acids as well. So this gluconeogenesis is a universal pathway which is taking place in microorganisms, plants, and animals as well. So we'll talk about first plants. So plants in the seeds, which are the growing organs for which will germinate into a plant. So seeds will germinate into a plant. You all know that. So seeds store nutrients such as proteins and lipids molecules. So these proteins and lipids, before attaining the photosynthetic activity, the lipids and proteins will be converted into a certain amino acids which may be a precursor of gluconeogenesis. So in the plants such as castor, so seeds are a storage organs of lipids. You all know that. So seeds store lipids in the form of a triacyl glycerols, you all know. So these triacyl glycerols will be converted to the precursor for gluconeogenesis, that is oxaloacetate. Then the other process will begin. So you may ask the question that glycolysis and gluconeogenesis, what is the difference between these two pathways? I can say these both are irreversible pathways, not irreversible. They are reversible pathways, I can say. But reversible pathways, but there will be three reactions. First reaction and second reaction, third reaction and final reaction, which is pyruvate to phosphoenol pyruvate. These are irreversible reactions in glycolysis. So these pathways require other enzymes for converting into a product. So in plants, the triglycerols are converted to oxaloacetate so that there will be preparation of glucose, and then glucose is converted to sucrose, because the main transporting molecule in the plants is glucose. So in this way gluconeogenesis works. Then we will come to animals. In animals, if we do, let's take the example of a human. You all know that glucose is stored in the form of glycogen in the liver. So the insulin hormone, which is very important hormone helping to store glycogen in the liver. So this glycogen, if you do any fasting or any exercise activity, the glycogen will all be used up, so that we will not have any energy for the body utilization. The very important minute thing is the brain. Our brain require 120 gram of glucose daily. So to maintain that if we do not eat anything, so our body itself will attain to gluconeogenesis pathway, so that the glucose will be maintained. In this way, the animals will utilize glucose for its metabolic activities. Okay. Then we will come to the pathway. You all know that same as the glycolysis, but here gluconeogenesis, it will run in the opposite direction. So I will say what are the sites where gluconeogenesis occurs in animals. In animals, majorly in the liver and in epithelial cells of renal cortex, there will be gluconeogenesis. And in the plants, the gluconeogenesis occurs in the seeds as well. So let's come to the topic. Here, glucose is a precursor for glycolysis, but here there are many precursors for gluconeogenesis. Mainly, we will take the example of pyruvate. So, lactate, it is present in the muscles, in the adipose tissue. When we have no energy, lactate will convert it into pyruvate. So, the main precursor molecule for gluconeogenesis is pyruvate. So, we want pyruvate. So, all the molecules should convert into pyruvate. Then, first I will discuss about the plants. In plants, we all know that triacylglycerol is stored in the seeds. These triacylglycerols will be converted to acetyl-CoA. Then acetyl-CoA is converted to succinate via glyoxylate pathway. This is the important pathway taking place in the plants. Let's discuss this pathway in the another video. So then succinate is converted into oxaloacetate. Not oxaloacetate, but succinate is one of the intermediate molecule in glycolysis. So succinate is converted into oxaloacetate in Krebs cycle, so that oxaloacetate also can enter the gluconeogenesis pathway. Then I will talk about the 3 irreversible reactions which are taking place in the glycolysis, so that gluconeogenesis pathway will begin. The first reaction in glycolysis, I will talk about first here. So the conversion of phosphoenol pyruvate to pyruvate is the irreversible reaction in glycolysis. So here it is very hard to convert pyruvate to phosphoenol pyruvate, because it requires more amount of energy to convert it. So how will you convert? Pyruvate is converted to oxaloacetate. Pyruvate first reacts with bicarbonate, then it requires 2 ATP molecules, and then it is converted to oxaloacetate with the release of PI, inorganic phosphate. Then this oxaloacetate, with the use of 2 GTP molecules with PEPS carboxykinase enzyme, it is converted to phosphoenol pyruvate. So it requires more amount of energy to convert. So now phosphoenol pyruvate we got. So like this only, as in glycolysis, the irreversible reactions occur. So now in these 2 steps, the conversion of 1,6 bisphosphate to fructose 6-phosphate requires another set of enzyme called fructose bisphosphatase. So the phosphatase means it is helping to remove the phosphate molecule from this molecule. So the enzyme phosphatase is nothing but it is removing the phosphate molecule from fructose 1,6 bisphosphate. So now we got fructose 6 phosphate. Then you can easily convert glucose 6 phosphate from fructose 6 phosphate. But here glucose 6 phosphate to glucose is very hard. So we require another set of enzyme called glucose 6 phosphatase. Here glucose 6 phosphatase means this enzyme phosphatase is removing the phosphate group from glucose 6 phosphate to convert to glucose. So in this way the gluconeogenesis pathway is running. Now glucose can be converted to sucrose in the plants because sucrose is the main transporting molecule in the plants. So now in the germination seeds, the glucose is utilized for their germination until the plants get photosynthetic activity. That means if until the leaves arises from the plants. So this is the idea of gluconeogenesis. So these three irreversible steps, glucose to glucose 6 phosphate, fructose 6 phosphate to fructose 1,6 bisphosphate, and phosphoenol pyruvate to pyruvate. In gluconeogenesis pathway, here these three reactions require high negative energy, very high negative energy. That means they are undergoing exergonic reactions. There will be high release of free energy. So these three reactions are highly exergonic reactions. And here in animals glucose is used up for our metabolism. So glucose can be converted to glycogen in liver, and if the insulin hormone is very good, so that our blood can utilize the glucose for our metabolic activity, and our brain will be good, which require 120 gram of glucose every day. So another important thing is in this topic is the triacyl glycerol is converted to acetyl CoA via beta oxidation. Let's discuss this topic later. Then beta oxidation, it takes place in the organelle called a special glyoxysomes called peroxisomes. So this beta oxidation in plants, I am telling about plants, but in animals it takes place in the mitochondria. That's another topic. So in plants, the triacyl glycerol is converted to acetyl CoA in peroxisomes. Then acetyl CoA via glyoxylate pathway is converted to succinate in glyoxysomes. Then succinate is the one of the intermediate molecule in Krebs cycle. So this is converted to oxaloacetate. Oxaloacetate is one of the molecule which cannot come out of the mitochondria. So here we require a shuttle system to come out of the mitochondria, because our inner mitochondrial membrane is highly specific and it does not have any specific transporter molecule for oxaloacetate to come out of the mitochondria. So here we are running a shuttle system, that is oxaloacetate with the help of acetyl CoA is converted to citrate. Then citrate, here mitochondria inner membrane as the citrate transporter, so that citrate can come out of the mitochondria to cytosol, and then citrate can be converted to oxaloacetate. So we can get in this way the oxaloacetate. This is called citrate shuttle system. It's a very important topic in biochemistry. So then it can come out of the oxaloacetate. Then it can enter the gluconeogenesis pathway in this way. Again, in plants, 3-phosphoglycerate is one of the intermediate molecule in the Calvin cycle. We can call this as C3 cycle also, or Calvin-Benson cycle also, so that it can come out of the chloroplast into the cytosol so that gluconeogenesis pathway can run like this. Ultimately, it should be converted to glucose, but in plants, it should be converted to sucrose. This is the idea of gluconeogenesis. That's it, students. I think I have covered all the idea about gluconeogenesis. Thank you.